I'll stop sharing at the end of the presentation and then we can go to questions from there. But my name's Judy Slape and I'm the Public Engagement and Involvement Officer at Health Data Research UK. And today I'm really happy to be giving a brief overview of HDR UK and the patient and public involvement and engagement that we do and how it shapes all of our work. And then really keen to open it up for any questions. There we go. So Health Data Research UK is the National Institute for Health Data Science, and our mission is to unite the UK's health data to enable discoveries that ultimately improve people's lives. To unite UK's health data, we must include the public, patients, and practitioners in everything that we do. So I'll start by giving a quick insight into what exactly we mean by patient and public involvement and engagement. And for this, I've also included communications. We use all of these different methods in our work. So to start off, communication is when we provide information and outcomes from projects, programs, and services to the public. So for example, we regularly share updates and information on our website and social media for patients and the public. But this is sort of a one-way interaction when we're just explaining something that we've done or that we're doing. Engagement is when we actively share and discuss information and outcomes from our projects and our programs with the public in an engaging manner. So for example, we hold public engagement events such as online webinars for members of the public to learn about and ask questions about our work. So you can go onto our website and onto our YouTube channel, and we actually have some recordings of some of our previous webinars on things like vaccine research for COVID-19, and you can have a watch of them. Finally, involvement shapes all of the work that we do, so across all of our programs. This is when work is undertaken with patients and the public, and they're actively involved in the development, running, and management of projects and programs. And I'm going to continue on this presentation, giving an outline of how we practice involvement. So there are a range of benefits of patient and public involvement. It's an intrinsic part of citizenship, public accountability, and transparency. In particular, involving patients and the public through the research process can provide accountability in ensuring projects are developed and implemented for public benefit. It can also improve public contributors' understanding of the use of health data, which can lead them to become advocates and critical friends. Crucially, active involvement can lead to more relevant and higher quality studies when implemented from the beginning and in a way in which it can influence and shape research. So this fundamental aim of, and I'm going to start dropping the acronym PPIE, which stands for Patient and Public Involvement and Engagement. So this fundamental aim of PPIE ensures research is relevant, acceptable, appropriate, and answers questions of importance to patients and the public, thereby avoiding research waste generated by studies that have little benefit to the public. At Health Data Research UK, we have a number of patient and public groups. So we have our Public Advisory Board who oversee all of the work of HDR UK, and then we also have specific delivery and steering groups with lay members. So we have a sort of overarching group known as the Public Advisory Board, but then on each lots of different projects and programs, we'll have specific sort of smaller groups of lay members to work with us. We also have a specific COVID-19 PPIE group and a HDR UK Voices group. These are networks of patients and the public who want to be updated about the work that we do, and also we promote involvement opportunities like one-off surveys or focus groups that we might be doing to this network. So our HDR UK Voices group, we are always looking for new people to join. Currently, we have around just over 80 people in that group. We also link to wider networks across all of our partners. So for example, we link with Understanding Patient Data and Use My Data as well. This slide just gives some examples of some of the Public Advisory Board achievements. So in 2019, we recruited the founding Public Advisory Board members. Since then, they've done a heap of work in helping to shape our work. So some examples is that they helped identify areas of greatest risk to public trust and data access. And they also shaped our COVID-19 approach and contributed to, we used to do weekly reports to SAGE, which we don't do weekly anymore. They went down to monthly, and now they're even sort of more infrequently. But at the height of the COVID-19 pandemic, we were doing weekly reports, and our public contributors were contributing to those reports. They were helping us to identify what was most important to the public. They've also been really instrumental in recommendations on data access process taken on by the Alliance and referenced in National Data Guardian guidance, which I will give some more information on shortly. Most recently, we recruited 7 new members to replace end-of-term members that had originally been recruited in 2019. So, as I just mentioned, the Public Advisory Board were really instrumental in recommendations on data access processes. So this paper built on findings of a recent survey of Alliance members initiated by Health Data Research UK's Public Advisory Board to gather information about patient and public involvement in the assessment of data access requests and the monitoring of data use. The survey was sent to individuals in 45 Alliance member organisations during February-March 2021 and revealed a wide range of approaches from full public involvement in decisions about data access to none. The survey revealed many missed opportunities to inform and involve the public about policies in relation to health data use, and only a few examples of excellence in public involvement and transparency. There was a lack of consistency in how data access requests are assessed and publicised. So it was apparent that most information about data access procedures is produced for researchers, not actually for the public. I won't go through the recommendations in detail in the interest of time. However, at each point, it was highly recommended that patients and the public are involved meaningfully. This was even more so in particular when data access requests contain sensitive and personal data. So I'm going to run through some examples of the impact of patient and public involvement during the pandemic. These examples are all from the Data and Connectivity COVID-19 National Course Study that we operate out of HDI UK. So working with patients and the public is a key priority to ensure that their voices shape and influence our work. Working together, we want to ensure that they influence our work through informing key decision-making, improving communication to ensure that it's engaging, accessible, and meets the needs of the public, planning and measuring patient and public involvement and engagement impact, working with us on challenging and complex issues that may affect public trust, and overall ensuring transparency throughout. This is a bit of a busy slide, but as the slides will be sent around to you later, I think it will be interesting to be able to give it a good read. But in 2021, we involved 1,297 patients and the public in the Data and Connectivity program. So that ranged from workshops, surveys, and focus groups to involving public contributors in our research funding calls, overall governance, and public engagement and communications. And I'll now go into 2 examples of the work that we've done with patients and the public. So the first example that I'm going to go into was a new clinical trial sponsored by AstraZeneca was established to assess if a particular course of treatment can safely treat outpatient non-hospitalized adults diagnosed with COVID-19 and prevent either severe COVID-19 illness or death. We worked with AstraZeneca as they looked at data-driven methods using data from test and trace to improve and accelerate recruitment of people who have very recently tested positive for COVID-19 into the study. Our patient and public involvement activity was to support AstraZeneca as they looked at ways to improve and speed up recruitment of people into clinical trials. And our activity focused on how acceptable the public would find an approach that would contact individuals who have had a positive test and have agreed to be contacted about COVID-19 research, as well as some information about data-related issues. We ran a really simple PPIE consultation and had 106 responses in about 6 days. Although the approach that's been taken to contact people to see if they would be interested in joining the trial has changed, it's still worth noting people's perceptions of how ethical it is for test result and contact details to be passed on to researchers at Oxford University to contact them about getting involved compared to test and trace getting in touch. People's perceptions were that they trusted researchers at Oxford University, and those who felt it was unethical to be contacted by test and trace for recruitment put this down to a few areas. Additionally, if it wasn't the researchers or test and trace contacting them to be involved, the only other source that people said would be acceptable would be for their GP to contact them about the trial. People preferred to be contacted by email, then by text, and then by phone, with common additional feedback being around phone calls could be a scam, and emails give people more time to digest things, but also they need to think about how to overcome digital exclusion. Additionally, the consultation showed that the majority of people would prefer to be given all of the information at once when provided with the test result compared to 48 hours later. When exploring this further, it showed that whilst they do want time to really digest the information and have the time to think about whether they would like to get involved in the clinical trial, they don't want to have multiple phone calls once they've been given a positive test. This was particularly the case if people were not feeling well after contracting COVID. It was also felt that by doing at once and being told about multiple trials at the same time, as long as it was brief and simple and signposted to additional information, it would give the individual more confidence in the approach rather than being a bit haphazard. I'm going to now move on to a second example of some work that we've undertaken with patients and the public. So the Data and Connectivity Program has undertaken two research funding calls, I should say. The first research funding call occurred in January 2021. At the time, we involved 3 public contributors to be part of the reviewing and scoring applications part of the research funding call. After this funding call was completed, we worked with those 3 public contributors, but also additional public contributors to develop best practice guidelines from our learnings. We had a whole range of learnings, right from how to actually put together a funding call application and initial selection criteria involving PPIE plans and benefit to the public in the researcher's application, right through to how we review and score applications, and also how we monitor how funded researchers are undertaking their patient and public involvement and engagement. These best practice funding call guidelines have been recently renewed after our second funding call when we've had additional learnings. I'll provide a link as well, actually, but you can also look up these best practice guidelines on our website. We have some top tips for successful patient and public involvement and engagement. Involving and engaging patients and the public as early as possible. Having clarity on what you'd like to achieve and tell that to your patient and public contributors. Resource and plan your patient and public involvement and engagement activities appropriately. Offer different ways for patients and the public to get involved and engage with your work. And be accessible and offer training and support. Document and record your patient and public involvement and engagement activities to measure impact. And most of all, be open to change and meaningful involvement and engagement, and be as creative and innovative as you can. So that's the end of my presentation, and we've just got our contact details there. But I'm really happy to stop sharing my screen and go to any questions.
Thank you, Judy. That was really helpful. So yes, if you have questions that you'd like to ask now, you can either unsilence yourself and also on video yourself and ask, or you can type it in the discussion on the right-hand side. As you've seen, Judy's colleague has put some more information in there with the links and some background information to what you just heard in the presentation. So if you have anything you'd like to ask or raise or discuss, then please do so now. If not, we'll have another opportunity at the end of tonight's session or indeed you can email us with any questions if you want to think about it and come back. Elizabeth, you have a question.
Hi there. Hi, Judy. Yeah, I just had a question. It was about something you said right at the beginning of your presentation, where you were talking about, I can't remember what the committee was called, sorry, but you said you recruited 7 new PPI members to replace end-of-term members. Just wondered what you meant by end-of-term members and what the rationale is for sort of potentially limiting, I guess, people to a certain length of time that they can contribute.
Yeah, definitely. So we have terms. I might, Sindhuja's on the line, and sorry to sort of pull you into the spotlight, Sindhuja, but you might be able to give sort of more clarity on our rationale for why we have end of terms.
Absolutely, Judy, and Elizabeth, thank you so much for your question. So this is for our Public Advisory Board, who provides strategic advice to the organisation. And all of our Public Advisory Board members are on a 3-year term membership. The reason we do this is to make sure that we can bring in some different perspectives, especially because the Public Advisory Board are so vital to guiding the direction of our work as an organisation. The other thing that we found is that when it comes to involvement in health data science, the patients and members of the public who get involved really end up knowing a great deal around how health data is accessed and used for research. And they're absolutely brilliant. In some ways, I feel like they know more than the kind of staff at Health Data Research UK because they're so dedicated to their role. But what that means is that sometimes they gain quite a lot of knowledge that they may not have that kind of, I'm going to say, person on the street kind of experience and knowledge and things like that. So that's another reason why we try to bring in fresh perspectives. But the outgoing members are so incredible and amazing to the work of Health Data Research UK that whilst they have stepped down, what we've done is kind of move them into different roles, and so that they're still a part of Health Data Research UK, but just in a different way.
Thank you. Yeah, that's brilliant. I like the idea of managing people's expectations there right from the beginning as well. Yeah, thank you. Thanks for your answer.
Thanks, Elizabeth.
If no one else has got a question, I've got a quick question. You spoke about the training and support that you give to your public contributors. Can you just talk a bit about how much training you give around the subject of data and data science, or is the training also about, if you like, general contributions and general learning for public contributors?
Yeah, definitely. Great question. So we have sort of different types of induction and training. The Public Advisory Board go through quite a large induction and training, which again, Sindhuja will know more about as she manages the Public Advisory Board. Yeah, I might just hand over if that's okay.
Yeah, of course, Judy. Great question, Rachel. Like Judy said, I think it kind of depends on the role that the patient and public contributors take on. So for our Public Advisory Board, because they are kind of across the whole organisation and therefore across a lot of different programmes, it is a bit more in depth. So as well as doing an induction around what is patient and public involvement and that kind of thing, we go more in depth around health data science as well. So kind of give them an overview of how is data accessed? How is data used? What are the safeguards in place? Where are the potential areas that could affect public trust? And so we've developed a health data guide, which, in all honesty, I would have really appreciated when I first started at Health Data Research UK. And it kind of goes through the whole process there. And so what we're actually doing at the moment is actually kind of taking information from that health data guide and putting it all up on our website so that it's accessible for anyone who is interested in finding out a bit more. So that's for the Public Advisory Board, but then I would say that for any specific programs, in addition to that, we would of course give the kind of background to the program and why it's important that we have patient and public involvement in there as well.
That's brilliant. Thank you very much. If no one's got any burning questions at this point, we'll move on because it's time to introduce our next speaker. But as I say, there will be time at the end, or indeed you can put the comments in the chat and we will answer those questions at the end or after this meeting. So thank you very much, Sindhuja and Judy. That was very interesting. And please feel free to join us for the rest of tonight's event. And listen to some of our other speakers. So thank you very much. Now I'm going to move on to our next speaker, who is Professor Guru Atal, who is the Deputy Director of the NIHR Nottingham Biomedical Research Centre, which is a partnership between Nottingham University Hospitals and the University of Nottingham. And the Biomedical Research Centre specialises in what we call translational research. So the process of taking scientific breakthroughs from the laboratory out into the research landscape to develop and test new approaches to disease treatment, management, and diagnoses. So Guru, I'm sure, will introduce himself and his background, but Guru also leads the gastrointestinal and liver theme within the Biomedical Research Centre. But this evening he's going to talk to us about an upcoming study, which, as you can see on screen, is called Demystify, and I will hand over to Guru and let him explain all about multi-organ fibrosis. Thank you, Guru.
Thank you, Rachel. I am a professor of hepatology. That's the only additional information I want to give so that you can see probably my interest in this project. I will describe how this study has come about and what particularly with regards to data this involves in an existing database as well as existing cohorts that we have got in Nottingham, and probably will describe the objectives and design of the study and some preliminary information that we have got on it. And after the talk, you can ask me any questions. Demystify Multimorbidity is a UKRI funded study, started nearly 2 years ago. It had three phases of development. Initial phase was applicants to propose potential research areas with some substance of what they might do, and then there is a review process where they shortlist some of the proposals with the small amount of funding where we can collate pilot data and build on the application, as well as most important is to build on a consortium of experts within the country to deliver the project. The topic was highlighted by UKRI that it should be focused on multimorbidity, and hence the background of why UKRI or funding bodies are interested in it. This is the data which has come from UK. You can see two sets of data here. One of them shows that over a period of time, we have gradually have people having more than one chronic condition at the same time. And this concept is called multimorbidity because historically, when medicine is taught, you're taught as a clinician to look at manifestation and then put it all together in one diagnosis with the idea that people are unlikely to have two conditions at the same time, so you should bring it together into one diagnosis. How can that be explained? This be explained. But now comorbidity, which is existence of two concurrent or more conditions in an individual, become more of a rule than an exception. We have 54% of people over 65 who have got more than one condition at the same time, 2015, and the projection is that it's going to become 68% by 2035. And the graph shows that gradually, in the different shades of colors here, people having more than 2, 3, 4, 5, and 6, and 7, and more than 8 conditions will be increasing. And this is, on the surface of it, will look like population aging, but it's not quite true. There are a number of factors, some of which we recognize, some of which we don't recognize. Hence, Chris Whitty, who is now Sir Chris Whitty, and Fiona Watt from MRC set out a call in Nature in 2020, and then followed by BMJ paper, which I'm cited here, to almost develop methodology of research for us to discover new patterns of comorbidity, which needs unbiased observations, as we are all trained to really look at one thing at a time, and develop training programs for clinicians to address this, because our training is to subspecialize and focus on what we know and then send it to somebody else to sort out other problems. So multidisciplinary approach to the delivery of clinical care as well as services. So research, training, as well as services had to change, and that is a call. On the right-hand side, you see the data on different age groups, and of course, there is substantial increase in multimorbidity, even when you adjust for a lot of factors which you think we know might be underlying it. Our proposal was to identify fibrosis or a scar formation as a phenomenon that underlies multiple organ inflammation, injury, and healing process. So you can see that inflammation is generated by persistent or recurrent injurious agents, which might be organisms, which might be risk factors we are exposed to, such as smoking or drinking or excess food or calories. And these develop inflammation. And if you think of obesity, most people consider that as a form of metabolic inflammation. It's low grade, so you don't really obviously fall ill to get admitted, but this slowly results in cell death in multiple organs at the same time. And the body's response to cell death is to localize it, because we are all used to pathogens coming in or organisms coming and infecting person, and the immune system goes there to try and localize this and then destroy it. But the process of meta-inflammation is so systemic that this process itself might generate scarring. Some tissue gets compromised. And if the injury and inflammation is persistent, then the compensatory mechanisms or defense mechanisms might themselves will persist and result in overall scarring in organs. So scarring can happen in the brain in a different way to other parts of the body called gliosis, but you can have it in heart, lungs, and liver, and pancreas, and most of the organs that I have mentioned. So we identified key organs which we know are interlinked, and the hypothesis was that there were some common pathogenic mechanism which links them beyond what we see as aging phenomena. And can we find out by analyzing some of the big databases that we have got, and then see whether that replicates in cohorts that we have got, and can identify signals early And when one organ is involved, can we pick up another organ developing ahead of time? And this is just one risk factor I have put in, and few other simpler risk factors I've already told. And we are looking for pathways which are beyond just simply what we know. Obesity is most explained for most people, and as I mentioned, excess fat is susceptible to inflammation, which is a metabolic inflammation, which is low grade and generates inflammatory signals, and individual organs respond to it. And if you look at obviously recognized pathways, one of them is what is called a short telomere syndrome. This is another concept of aging, but in this condition, there is premature aging. If you think of standard aging, every time cell duplicates, the cap of the chromosome gets broken down a little bit, and body has a mechanism of repairing it. So you could argue that our chronological aging, standard aging we all go through, can be detected by how long this telomere cap is in the chromosome. But if you are exposed to risk factors, say for example, you're overweight or you smoke, then there is extra stress put on the body and the cells, and that really generates much less defense mechanism working, much more aging. And there are studies to show that if you measure the telomere length of people who have risk factors like smokers who are overweight compared to the same biological age, a 50-year-old, if you compare one with the smoking and obesity and the other one without those risk factors, telomere length can calculate your biological age, which might be almost 10 years different, which comes up, this is a concept that there may be some genetic signals might detect premature aging. And these are called telomeropathies, and these are recognized. These are seen as rare conditions. One of the rare condition is a specific variation or mutation in a telomerase, an enzyme which helps in repairing this cap being broken down. And if you look at that kind of cohort, you see as you go along, telomere length shortens, and more shortening telomere length is, more organs manifest. So they might initially manifest with some bone marrow manifestations, and then you might get liver diseases, and then you get pulmonary fibrosis. And if they come to different specialties, and one will be focusing on it on their own, but there is no preparation when they come to pulmonary fibrosis, and they end up with disability and very rapidly die. So our thoughts were that if you look at a big database, we might find out non-syndromic, in other words, unrecognized variations in telomere length we might find, or we might actually find a pathway which we can use and pick up this kind of multi-organ scarring early, and then use it to help people by diagnosing it earlier, and then have treatment or interventions which are readily available. So our objectives are to identify clusters of multi-organ fibrosis and find out how common they are, and find out risk factors associated with them, and then see what happens in the long term for them. What kind of sequence in which more serious events develop, like failure of organs, lung or liver or other organs, and needing major interventions like transplantation or leading on to death. Then we wanted to identify biomarkers or signals which might be genetic. I just gave you an example of, or simpler available tests that might pick up, so that you can get a trajectory. First of all, try and prepare a pathway of which organs come first so that other organs can be looked at, and at least we can reduce the number of organs being involved or slowing the development of fibrosis. We were lucky enough to be one of the, I think, 11 consortia who are funded. This was led by Gisli Jenkins, who is a lung fibrosis specialist who is now in Imperial. But I am leading this project from a Nottingham behalf. I'll come and tell you the project group in a minute, but this is the first step. So large database, UK Biobank, if you go and put in codes, which were the data science and its expertise comes from, you put in codes, specific codes of outcomes or disease codes, you get the number of cases you can see on the screen. Circulatory system comes up with hypertension as an outcome. So you can argue whether it is really in all cases so solely dependent upon fibrosis or not, but that's the commonest codes that come up. But more closely look at about 1,528 patients with liver disease come up. Same with the lung, pancreatic, reproductive organ. Again, there is a local fibrosis develops due to ovarian and endometriosis-like conditions. So you identify these people and then look at genetic signals. I'll show you some of the data, preliminary data, if I can, at the end of this. And there is initial factors are that if you do a genetic course, it looks like lung fibrosis and liver fibrosis correlate with more than one organ having fibrosis quite closely. So we see that there is some plausibility for our hypothesis that more than one organ fibrosis seem to exist when you look at in a database, a big database like UK Biobank. Our objective is to then see whether it exists in our cohort. So these are the cohorts that we have developed over a period of many years. We were lucky to also have a biomedical research center for the past 5 years and digestive diseases group, Nottingham Digestive Diseases Center. That's how we started. We're even luckier to have NIHR Biomedical Research Unit, which we started in 2009. So from 2009 onwards, we have been focused on a number of different conditions thanks to NIHR, and we have now accumulated large group of people who have very, very well characterized fibrosis. These are patients, many of them have had a biopsy confirming they had liver fibrosis. So the study design has 5 different large cohorts. I'm showing you about 1,600 people with liver conditions and how they were derived through different kinds of studies. In the same way, at the bottom of here, there are pancreatic diseases. And again, another 1,500 people. And inflammatory bowel disease, Crohn's disease, there are significant fibrosis and it causes major ill health. So we have another 7,000 people who have got Crohn's disease from UK Bioresources. It's another national source, of which 1,000 people are from Nottingham. In the same way, Gisli Jenkins, while he was here, he developed a large cohort of patients with lung fibrosis. And we have got Nick Selby, who is a clinician in University of Nottingham, clinician scientist, professor now, and he works in Derby. He's got another 2,000-3,000 people with chronic kidney disease. So we start with 5 primary organ fibrosis and then look into their data that we have collected and see this is just a code-based diagnosis. So I'm only showing you the codes which are relevant to liver-related fibrosis. In the same way, we have got 28 different outcomes, and each of them are described with number of codes. And in the cohort that we have got, you start from the primary organ cohort and then find out what other organ fibrosis codes they have acquired during their follow-up. from the starting point of developing the cohort. Then you pick up, our aim is to pick up about 2,000 people who have got definitely during their natural history and follow up that we have got multi-organ fibrosis, and then see the signals that we have identified from UK Biobank replicate in our cohort, which means that we have confirmed that there is enough strength in the hypothesis. And many of the studies, just to say how the disease cohorts develop and how it progresses. So I showed you some of the studies where we had previously enrolled patients from, and we had involved patients to develop these cohorts, and with their permission we developed ethical application, and with their support we consent patients to get blood samples and store them, and many a times future studies. And this is really important for patients, because when you bring them once, you just don't want to really ask for a hypothesis unless they are very keen that only sample is used for that purposes. So liver cohort, for example, have got already over 5,000 people who have consented for their samples to be used for future research. So this is the time we can use their samples to test the hypothesis, is that whether these patients have likelihood of developing further scars. And for example, chronic pancreatitis patients, we haven't got such cohorts of samples. We are now applying for ethics to get consent to invite those patients to come back to us and then give blood samples if they are willing to do so. So we will aim to identify 500 people who have got 2 or more organ fibrosis in each of the 5 cohorts I have mentioned. So you will end up with 2,500 people, or at least 2,000 people, where we can test the hypothesis that we have developed from a UK Biobank. This is the genetic analysis. This is very preliminary, and I have taken away the results because they are not robust and double checked or triple checked, but explain to you what is what we call a genome-wide association study. As with data science, these are hypothesis generating methodologies of research. And there are advantages are that you're not looking for a thing which you think is going to be true, and which means that in a way it will avoid even a human bias as a scientist you might have. The disadvantage of it is that there are so many things that might occur by chance, which means that you need to have a statistically robust method to make what you identify as a positive signal to be robustly positive. So this is one such methodology called Genome-Wide Association study. In this picture, we have got 1 million dots, but they are all overlapping so that some of these look like bands for you. But the dots separate out more and more when they are exception than the rules. So along the x-axis is different chromosomes and number of variations of the chromosomes which are really identified. On the y-axis, you have got log of p-value, which means that if your p-value is lower, the dot which comes higher. And here is a red line, which means that a p-value which tells you what's the likelihood of certain signal or finding being by chance. So lowest chance is better to be considered more authentic bona fide signal. So this is a cutoff. It's about, I think, 10 to the power minus 8. It looks almost like 7.5 or so, but I have double checked it. It's meant to be 10 to the power minus 8, which means that if you have got dots above that line, these are likely to be authentic signal. So these are the preliminary signals that we see, which are identified by the preliminary analysis that we have done in a biobank as signals that are common in people who have got more than one organ fibrosis. I told you how we defined it in UK Biobank previously, which means that maybe there is some plausibility in the hypothesis. That is one of the signals. This is another thing we are going to look at. I told you that we are looking for signals or biomarkers that can identify fibrosis or scarring in multiple organs. Nottingham with Susan Francis and Penny Goland and few other radiologists. Dorothy Orr actually is also an applicant, have got expertise in looking at fibrosis in multiple organs, which is gliosis in the brain, but fibrosis in all the other organs. This is some of the data that we have extracted from our previous studies. I won't go through the details except to say that why this kind of technology is important. So if you do an MRI scan, you focus scan, and you optimize your protocol to look at the liver or a kidney or heart, depending upon what you want to do. So this is specifically designed protocol, which can be done in as much time as we can in a clinical MRI scan, but designed to pick up scarring in multiple organs. So if you have that, and if you think of, say, someone comes with liver fibrosis and you want to really identify this person who might develop other organ fibrosis or might have already have silent or asymptomatic fibrosis in another organ, if you want to really have a head start so that you can produce some interventions which are effective at preventing getting another organ fibrosis, then one scan will be able to pick it up. That is another hypothesis we are going to test. And what do we do by identifying it? This is unrelated to the demystify, but we have already got an ethical approval and we have started enrolling to this study called research study. And this is now focused just on liver fibrosis, which is targeting a pathway called mTOR, which is a signaling pathway, which is a generic pathway actually. And we already have a medication which we use for stopping transplants rejecting called sirolimus, which can be used in a very low dose to stop fibrosis, or that is the hypothesis we are testing. So if it worked, then you already have a drug which can be repurposed with a low cost, without development, accelerating translation to prevent people getting fibrosis in other organ. It's just a concept. This is a proof of concept study, but that is what we hope the best outcome from such a thought process is. Just to kind of highlight that we are biologists or focused on biology, we do studies on biology, but a lot of other factors which we may not have expertise in also influence multimorbidity. This is from the same paper which I cited in my first slide from Sir Chris Whitty and Susan Watts, which shows that just education or lack of education or degree of education seem to determine multimorbidity, which says that all we need to do is to educate people more. Thank you very much. Any questions? I'm happy to answer. Thank you.
Thank you, Guru. That was very interesting, and I think a lot of, pardon the pun, food for thought in terms of what might be ahead of all of us without that awareness and education. In the interest of time, I'm just going to move on to our next speaker, and then if there are any questions, if you want to just type those in the chat, and we'll come back to them at the end because time's moving on. So our final speaker for this evening is Fiona Pierce. Thank you, Fiona, for coming along. I know it's not you're another one who's in fashion with the COVID. So Fiona's going to talk to us about the Recorder and Melody study, which is looking at rare disease and how they're using data to inform that research project. So welcome, Fiona, and over to you.
Thank you very much, Rachel, and hello everybody. Thanks for the opportunity to speak to you this evening. I hope I'm sharing my slides, but can you let me know if you can see them?
Yes, you are, and we can see them.
Lovely. So what I'm going to talk about tonight is just an example of research that we're doing at the University of Nottingham using health data in action, health data research in action to try and help improve the health of people in the UK. So, yes, so I lead a project which is an HDR UK adopted project. So this is funded by the NIHR and has been adopted by the Better Care theme at HDR UK. So I'm part of the HDR UK fold. So what I'm going to talk to you about tonight is rare diseases and how difficult it is for people with rare diseases and people trying to do research in rare diseases, because essentially, because they're rare, each rare disease affects fewer than 1 in 2,000 people. It's really difficult to get enough information together to know anything about them. But there are so many rare diseases, more than 5,000 different rare diseases, that when you take them together, rare diseases are actually very common. 1 in 17 people in the UK will be affected by a rare disease in their lifetime. That's 3.5 million people in the UK currently affected with a rare disease. And the problems are huge. Health outcomes are poor, much worse than people with an equally severe common disease because of all the difficulties of doctors not knowing what it is, people not knowing what it is, not knowing what the best treatment is. And research is difficult, meaning that often for rare diseases, there is no evidence-based treatment. So health data, why and how can health data help in this? So I think big data are essential for rare diseases. The traditional paradigm of doing research is to do it in a single centre in a single hospital. But if you take that to a rare disease such as Takayasu arteritis, which is a inflammation in blood vessels, which I'm researching at the moment. If you try to do research in Takayasu arteritis in a single centre, a single hospital, you'd expect to have fewer than 5 people with that disease, and they, of course, might not all want to take part in your research. And even if you go for a multi-centre approach, getting several centres from across the UK, you'd still probably end up with fewer than 100 people. So the approach we're taking is to use health data for the whole of England. And I'm really excited that for the first time in Takayasu arteritis, we've been able to find people from across the whole of England. And we're currently launching a piece of research with nearly 1,000 people included. But before anyone feels too alarmed, I'll just show you a little bit about what the health data I'm talking about is. So I'm not talking, we don't have access to anything confidential that you might discuss with your GP or with the doctor when you're with them in a clinic visit. What we get is really very sparse but incredibly important data that's routinely collected as part of the administrative processes of running the NHS. So the data I'm talking about, this is mock data because it wouldn't be right to show you real data, but this is an example of the sort of data I can see. So each person has a HES identifier. That's a number that is unique to them in the hospital episode statistics. That's the record of hospital admissions. You know, and I've put this number in here. You can see that it's, you know, the numbers 1 to 0 in ascending order up to 9 and then back to 0. But I think what is important to say is that although this is technically personal identifiable information, in that there is a key that means that somebody could tell you who this is, whose mum or auntie it is. As a researcher, I have absolutely no idea that, you know, this could be anybody. This could be my auntie in Surrey. This could be someone I've never met up in Northumberland. It's sort of, so there isn't anything, I think, terribly intrusive about this sort of data, but it's incredibly useful. So what I can tell from this line of data is that this person has Takayasu arteritis. That's the 314 code, but also really importantly, X921 tells me that they've had an infusion of a high cost immunosuppressive drug, which is approved, and it's one of the very few evidence-based treatments for this incredibly rare disease. And they had a stay in hospital between the 1st and the 6th of February 2020. So how do we do research in rare diseases? Well, back in 2016, Public Health England created a new arm of national disease registration to join the National Cancer Registration Service, which has been around for the last 20 years and has really been instrumental in improving outcomes for people with cancer in the UK. So this new part of it is looking at congenital anomalies and rare diseases and offering the opportunity to register them in a systematic way across the whole of England. So the research group I work in Nottingham, we work in collaboration with the National Congenital Anomaly and Rare Disease Registration Service, who were at Public Health England until it was dissolved last October and have now moved into NHS Digital. And it's a service designed to support clinicians and patients, service delivery, commissioning and public health. And through the National Disease Registration Service, we have access to the sort of healthcare data I showed you in the previous slide for the whole of England. And I lead what's called the Recorder project at the University of Nottingham, and the aim of our project is to work with the National Disease Registration Service and find ways to register people with rare autoimmune rheumatic diseases. So that means essentially finding everyone with these rare diseases in England and creating a register, so like a school register, essentially a list of names and NHS numbers, so that there are opportunities should people wish to take part in research for them to do so, but also for the sort of surveillance that the NHS does to be able to be done in rare diseases for the first time. So these brightly coloured, weird-looking words on the left are the specific names of the rare autoimmune rheumatic diseases. But what do I want you to take away from it? Not the specifics of the difficult to pronounce, but that they're serious diseases. They're all caused by the immune system attacking the body, and they're all treated with drugs that suppress the immune system. But we know almost nothing about them. We don't know how many people in the UK have them, what the risk factors are, what happens to people with them, because there's been little or no research in any of them. So back in in January this year, the UK published its Rare Diseases Framework. This is the Department of Health and Social Care setting out a policy document of how over the next 5 years, the UK plans to deliver better health outcomes and improve the lives of people living with rare diseases. So this was published last year, and then there was a response to it published last month. And I'm really proud that our research in Nottingham was included in the policy document. Here we are, the registration of complex rare diseases, exemplars in rheumatology, the Recorder project, is mentioned specifically as one of the case studies for England of work that's going on to support the framework. And here, so it's a collaboration between NCARD and the University of Nottingham. So we'd set that up and we'd found and started to register people with rare diseases, with those rare autoimmune rheumatic diseases, just as COVID-19 came. And COVID-19 was an extraordinary and unprecedented situation for everyone in the world, but particularly difficult for people who with suppressed immune systems. And as you can see on the right here, the guidance for clinically extremely vulnerable people came out very early in the pandemic, at Easter 2020, and people were advised to stay in their own homes and not leave under almost any circumstances other than to go to their own health appointments, because of concern that people with suppressed immune systems would be at increased risk from COVID. What we did through the register that we had just created was to see whether people with the rare autoimmune rheumatic diseases who hadn't been included in the shielding information because nobody previously been able to find them, we tried to work out if they were in fact at increased risk from COVID. So we used hospital records and that's the same data I showed you earlier, from the whole of England, looking at data from March to August in 2020, to see whether people with these conditions were at increased risk of having an infection with COVID, being hospitalised with COVID, or sadly dying of COVID. And what we found was that compared to the general population, for people of the same age and sex, people with rare autoimmune rheumatic diseases were more likely to have infections. For every 2 infections in the general population, there were 3 in the rare autoimmune rheumatic disease population, but also were more likely to die from COVID. For every 10 deaths in the general population, there were 24 deaths in the people with rare autoimmune rheumatic diseases. So they were 2.4 times more likely to die of COVID than people of the same age and sex. So we were able to feed this back to NHS Digital, who were running shielding, and get letters out to those people. We were also able to think about what else we could try to do to improve health and the advice to people with rare autoimmune rheumatic diseases. So we put our findings together with other research groups who'd found that people with rare autoimmune rheumatic diseases and suppressed immune systems also don't respond as well to vaccination against COVID-19. So we began to realise that people were essentially in double trouble. They were not only more likely to have severe COVID if they caught COVID, but also were less well protected by vaccination. And we thought at that point, we really, really, really need to do more here. We need to find out who may benefit from treatment to prevent severe COVID illness. And we applied with a group led by Imperial College to the MRC for a national study to invite huge numbers of people to test antibodies to see whether they've got antibodies following vaccination that will protect them against COVID-19. So we were able to use that register that we've built using the national data. We've been able to write to 20,000 people with vasculitis, lupus, myositis, and scleroderma to invite them to say, would you like to take part in a study that you can do in your own home that allows you, by a finger prick blood test, to check whether the vaccinations you've had will protect you against COVID-19. And that study's underway at the moment. We've posted out the invitations and people are taking part. And it enables people to find out if they've got antibodies, in which case I think people will feel relieved, because many people have been very concerned about whether they would respond to vaccination. But also for the people who find they haven't got antibodies, they will then be aware that they can access additional COVID-19 treatments should they develop COVID-19. So through the rapid changes over the last month or two with the government's response to living with COVID, they've brought out new guidance at the end of February for people whose immune systems mean they're at higher risk. And that includes offering new antibody and antiviral treatments to people, particularly those who haven't responded to vaccination. So this is the study page for the MELODY study. This is the study for people with suppressed immune systems to find out if they've got antibodies. There's a QR code here if anyone wishes to scan it to get to that webpage to find out more about the study. But in summary, I hope that in this whistle-stop tour of the last few minutes, I've taken you through how research that we're leading here at the University of Nottingham and in collaboration with Nottingham University Hospitals Trust has used health data through research to find people with rare autoimmune rheumatic diseases and who have never been able to take part in research before, and invite them to a study that is current and relevant and hopefully will help people know what to do if they catch COVID-19. So here are my acknowledgements. I'm part of a wonderful team who are taking this forward at the University of Nottingham. We work closely with the National Congenital Anomaly and Rare Disease Registration at NHS Digital, and I'm ever so grateful to the funders who have been funding this work. So thank you very much. I will try to stop screen sharing if I can find the right button.
That's fine. Fiona, thank you very much for that presentation and for the important work that you're doing, as you say, for that population of people who early on in the pandemic, I think, were very worried and often felt very forgotten by what was happening to the general population. While you were talking, we have a question in the chat for you, which has come from Guru. I don't know if you want to ask your question, Guru, but I can read it out. So what he's asking is rare disease definition based on the genetic variation that underlies the disease or population prevalence of that disease?
Thanks, Guru. So in this instance, the rare disease definition is the one that was created by the European Union, which is any disease with a prevalence in the population at risk of less than 1 in 2,000. So if fewer than 1 in 2,000 people or 5 in 10,000 people have it, then the disease is considered rare, and that enables the legal permissions to look at the health data for the whole of England by working with the National Congenital Anomaly and Rare Disease Registration Service.
Thanks for that answer, Fiona. So just a couple of minutes now, if anyone does have any further questions for Fiona or for Guru or for our colleagues from HDR UK who spoke earlier, please ask away or type away in the chat if you would like to ask a question. And Elizabeth, I think you have a question.
Yes, I've got a question for Fiona. Thanks, Fiona. That was really, really interesting. I just wanted to ask about the 20,000 people that you've said you contacted, and sort of what sort of rough percentage of people are taking up the offer of being involved in the research. And then sort of tying that back, my second part of my question is tying that back to what Judy was saying earlier, that when they sort of did the research they were doing about how people like to be contacted, they found that it was sort of through email rather than being telephoned or written to, and that they'd like to have all the information right from the beginning. So I just wondered what your sort of methodology is for contacting people, and whether you give them all that information right from the beginning, and if they don't respond, whether you have any follow-up mechanism to try and get them to be involved.
Thanks, Elizabeth. And like you, I listened with great interest to Judy's presentation at the beginning. So we've been writing to people, sending letters to them at home, and that's very, very interesting that people would rather be contacted by email. Unfortunately for us, we don't have email addresses. They're not routinely collected by the NHS, so we couldn't access email addresses, which was regrettable, because I think that it's interesting to know that would have been a preferable way of doing it. I think the next part of your question, Elizabeth, how many people have responded? We've had about a 25% response rate so far. So 1 in 4 people that we've contacted has chosen to take part. And we've written to people and sent text message reminders. It's interesting. I think that perhaps, and I don't know, we thought maybe we would get a higher response rate. We've been perhaps a little disappointed that the response rate hasn't been higher. And I wasn't sure whether we have just been in some ways unlucky that we've launched the study at the same time as the government have said that COVID's over and doesn't matter anymore. Because I think it does matter. It still matters a lot to a lot of people. And there was another part to your question, Elizabeth, and I'm trying to think what it was. Do come back in and remind me.
Sorry. Yeah, no, I totally agree with you about that. Maybe the timing as well, and it is regrettable, isn't it? That just as you're sort of starting all this great work, it's gone. It's gone now, COVID, hasn't it? Which obviously, ludicrously, it hasn't. I just wanted to know what information you sent to people, whether it was all there right from the beginning and sort of laid out for them.
Yes, brilliant, thank you. So yes, so we tried to make all of the information available from the beginning. The letter comes with a sort of a couple of paragraphs on the front, just sort of introducing the study in plain English, and then the sort of next level of information is a set of frequently asked questions on the back, and then it links through to the Melody Study website, which is the same one that I showed the front page of on one of my slides, which has all of the detailed information. So we tried not to overload people in the sort of initial communication, but as you say, all of the information is there at that time point should people wish to access it. And again, Judy, I was very interested to hear that you've done research on that and to know that that was that was what people's preference was.
Yeah, I mean, I'll definitely note that that was people's preference, particularly when they had been diagnosed with COVID-19 in those initial couple of days. And I guess it was at a point of COVID as well, quite a while ago when there was, a huge amount of worry and stress, obviously. So I think also it's that those heightened moments in the first 2 days of a diagnosis of COVID, how would you want to be contacted?